Hello and welcome to another lecture. So this lecture is cellular respiration, and we're going to split this lecture into two. Today's first lecture is about glycolysis and the TCA cycle. So like every time, let's recap what we discussed in the previous lecture. There we talked about chemical bonds in general. We talked about ATP. We talked a little bit about enzymes. So we said that chemical bonds can store and release energy. So the energy that is in a molecule is really what is stored in between the atoms in forms of this bond that is made by electrons. So the chemical bonds can store and release energy. ATP is the universal energy storage molecule. So that is the molecule that just through evolution, trial and error, if you will, has proven to be a molecule that works really well for being made and being broken down and made and broken down, and in this process either storing or releasing energy. Energy costing reactions can be coupled with energy releasing reactions, so you can use the ATP breakdown while you're doing another reaction, and that energy that is transferred can be immediately used in the reaction that requires that energy. We said that enzymes reduce the activation energy and allow reactions to occur in living systems without high energy input. And there we had talked a little bit about the different types of reactions, endergonic, exergonic reactions. And we had said that the enzymes can reduce that bump that you need, that first hump of energy that a reaction has to go over in order to then release energy. And so that's the activation energy, and enzymes can reduce that. Enzymes contain an active site. That's where everything happens in the enzyme. So everything else around it is just sort of holding that active site. And in the active site, that's where we said that's sort of a key and lock mechanism where the substrate, that is the thing, the molecule, that the enzyme works with, that's where it docks onto the enzyme. That's called the active site. And we said that the three-dimensional structure of enzymes is determined their function. And of course, that sort of led us back to think about the types of ways to describe a protein with its primary and secondary, tertiary and quaternary structure that make up the three-dimensional structure of this enzyme. Enzymes can be inhibited, for example, by their own product. All right, so that brings us to what we're talking about today. So let's think about cellular respiration from the big picture first. Your book has this graphic, and I want to walk you through this graphic so that you can sort of, look and think about cellular respiration from the 30,000 feet level, first to see what's going on there, and then we're going to break this down, and in the end, we're going to go into the molecular nitty-gritty. So we're starting with carbohydrates. Oftentimes that's glucose. It could be different sugar. Oftentimes these different sugars, if those are the ones that are used, are first modified so that we have glucose as a starting molecule. But let's just say carbohydrate, a sugar molecule here, is broken down via catabolism, right? The metabolism of breaking stuff down. And in the end, what we have is carbon dioxide. So that is, there's the carbons here, there's the oxygen there. And the hydrogens, remember that carbohydrates is carbon with water around it, so carbon plus H2O. Well, those H2s, those are going to be plucked off one by one, and they're going to take the electrons with them. That all requires enzymes for that to work. And they go through something that we here just simply call an electron carrier. And I'll show you the molecular structure of that a little bit later. We'll talk about electron carriers. They take the energy with them that later on will become part of this ATP molecule, of many, many ATP molecules, in fact, through a process that's called oxidative phosphorylation. And in that process of oxidative phosphorylation, lots of ATP is produced, and electrons are sort of moving around they're transported from one place to another, if you will. We'll talk about this part in the next lecture. That is called the electron transport chain, and that culminates, it ends in the formation of water and the use of oxygen. So cellular respiration requires oxygen, and it produces water, It requires carbon dioxide, and it also produces carbon dioxide. It requires carbohydrates, and it produces carbon dioxide. And then there's this other way of producing ATP, and that's called substrate level phosphorylation. And in that part, a little bit of ATP, not too much really, but still some, is produced, and that does not require oxygen. So some organisms that don't use oxygen actually still use many parts of this pathway, but they don't use the part that requires oxygen, and they produce all their ATP through substrate level phosphorylation. We'll come back to that at the very end of the next lecture, not this time. So all of that together gives you sort of the big picture of cellular respiration, what we start with and what we make. So what you should remember is that the input here is glucose or carbohydrate and oxygen, that the output is ATP and carbon dioxide, and that there are two types of transfer reactions that are referred to as substrate level or oxidative phosphorylation. So oxidative phosphorylation requires oxygen. The substrate level does not require oxygen. Okay, so we will need to talk about some chemistry. All of these steps are really classical biochemistry. And if you are interested in biochemistry at some point, these are the kinds of reactions that a metabolic biochemist thinks about all the time. So we need to think about what happens in these reactions in general. And many of these reactions are called redox reactions, reduction and oxidation reactions. So let's think this through step by step and sort of see what's happening when sugar gets burned or really used up in cells to produce all this ATP. Let's start with this summary reaction. So a chemist would write this down as the summary reaction. This is the formula for sugar, C6H12O6. And again, you can see, if you think back to one of the very first lectures where we talked about the molecules, the biomolecules, macromolecules, we also called them, large molecules that have carbon and then water around it, right? You can see this is H2O, really, H12O6. So it's 2 times as many hydrogens as oxygens. And each of those is around a carbon. So that's why it's called a carbohydrate, right? It's hydrated carbon. When that gets fully burned with the use of oxygen, then what you get in the end is carbon dioxide. So that is the stuff that we exhale. That's the stuff that's produced when something is burned to completion. And water is produced, and energy is released. And so that is the energy that the cellular respiration, of course, is after. So I want to show you really what happens when sugar burns, because that's a really nice way of looking at it. This is, of course, not what happens in the cell, but if you have sugar and you burn it, you're basically producing all of those parts that I just showed you, even if you just do it in the lab. So let's listen to this little YouTube clip here. So here's a test tube, and it has sugar in it. There's some glucose at the bottom, and now you're adding energy, right? Remember that you need energy, activation energy, to get any reaction started, or most reactions started. So the energy comes here in the form of heat, a flame. Of course, in the cell that would be different. But now you can see that the sugar starts to melt. It starts to react. It gets brown, right? Almost black. Of course, the black part there is carbon. It's just carbon. You may wonder, why is it carbon? I just told you it produces carbon dioxide and water. And that is true. You can see that there is condensation starting to form here on this test tube. Later on, you will see a little bit better how much water and carbon dioxide comes off. But the reaction in the lab never comes to full completion. So what you leave, what you're left with here is some residual carbon, basically just black carbon. Okay, now you can see, you can even hear some, the reaction relatively violently giving off this steam which is water, steam, and carbon dioxide comes off. And then in the end, if the sugar is not completely burned, you end up with a rest product, an incomplete reaction, and that is then just carbon. Okay, so how does that relate to reactions that go on the cell? Clearly, we don't have a little Bunsen burner in each cell, and clearly we're not producing a bunch of carbon that's left in the cells. So why is there black carbon left in this demonstration? It's because the reaction doesn't go to completion. It's because there's not enough oxygen available to produce only carbon dioxide and water. But you did see that carbon dioxide and water in the form of some sort of a steam is released. And you also saw that energy was required to get this reaction started. And so if you think about this from the point of view of an exergonic or an endergonic reaction, the way that we discussed it earlier in this class, we looked at free energy, or Gibbs free energy. Gibbs is the guy who thought of this. That's why it's referred to as G, delta G. Sometimes we talk about plus delta G, minus delta G, right? The energy that's in a system that can be given off and used for work, in this case that we're interested in the end, to produce ATP. So if you look at this reaction scheme here, then we have a relatively high starting energy, and then in the end we have a relatively low end point energy, and these are our products. This is our reagent at the beginning. So sugar, carbon dioxide, and water produced, and then we can connect this with this bump here. That's of course the activation energy that's required first, and then all of this from here on down is free energy that comes off that can be used for work, and the work that it's going to be used for in the cell is to produce ATP. Now, you could do this in one step. You would release a lot of energy all at once, and with that a lot of heat, and that could cook the cell, I just sort of say here. Of course, that doesn't happen in the cell. That would not be very useful. So in the cell, it really looks much more like this, where you have a series of small steps, where each of those steps releases a little bit of energy, and that energy can then be captured in coupled reactions. Right? Remember that these reactions can be coupled so that one energy producing or releasing reaction can be coupled with one that is requiring energy, and so together they can pass that energy from one to the other so that that reaction can work. And that's what's shown here in this diagram of these multiple steps, all of these different steps that where the product has a little bit less energy than the original reagent. And we'll look at many different chemical steps, chemical reactions, where this kind of energy transfer occurs. Okay, so what you should so far remember for the big picture is that energy from burning food is harvested in small steps, and that happens in mitochondria, also in the cytosol, and then transferred to ATP. That hydrogen from food molecules is ultimately transferred to oxygen, which forms water and is then exhaled. Carbon from food molecules is exhaled as carbon dioxide. So when you exhale, and you can certainly see that in the winter when you're outside, and you just sort of exhale deeply, or you breathe against a mirror or a piece of glass, you see that there's condensation on that glass or on that mirror. And that condensation is water. And that water is the outcome of cellular respiration. So that is the water that comes out of our mouth, essentially, out of our lungs, is the waste product of the burning of food molecules, sugar molecules, through cellular respiration. And then the carbon dioxide is also exhaled, and you can't see that the same way that you can see water. So I write here, the term cellular respiration comprises the process in which the energy from exergonic metabolic reactions is transferred to ATP. Okay, so how does this energy transfer happen in chemical reactions? So we want to spend some time looking at energy transfer. I've been talking about how energy is in the form of electron containing bonds, chemical bonds. We talked about all kinds of different bonds. And so we need to spend a little bit more time thinking about energy transfer through electrons when chemical reactions occur and when these electrons change their binding partner, essentially. So that is what happens in so-called redox reaction. Most reactions that are involved in respiration, in cellular respiration, are redox reactions. And so this is one thing that you need to learn in this chapter, what a redox reaction is, and also how it relates to cellular respiration. We will see that redox reactions also are making up other very important reactions that make up life. So we'll later on talk about photosynthesis. Those are also all redox reactions. So it pays off now to learn what a redox reaction is. Okay, so bear with me. Here's a chemical reaction where you have sodium and you have chlorine. And so this is just elemental sodium and elemental chlorine. You would never do this reaction in a lab, really. This is very nasty compounds that go in there, chlorine gas and sodium, very reactive. But when that reacts with each other, you know already from the first two lectures maybe, that the valence shell of chlorine has 7 electrons, and so it requires, or would like, whichever way you want to look at it, an extra electron to have a full valence shell of 8. Right? 8 valence electrons create a very stable electron environment. And so the reason why the electron goes from sodium to chlorine and not from chlorine to sodium is twofold. One is that it already has 7, just requires 1 more, so that it has 8, while sodium only has 1, and so it can give off that 1 and then is left with the next lower shell, which then it becomes its valence shell, and then that is its outer shell, and that is then full, and so then sodium is happy as well, is also stable. But the reason for why that also happens, of course, is electronegativity. Right? We talked about electronegativity as one of the properties of every element, and chlorine being at the very top right hand of the chart, the periodic chart, is very high in electronegativity. Sodium is very low, and so therefore electron moves from the sodium to chlorine. That means that sodium in this process loses an electron, and chlorine gains an electron. So now, by definition, every reaction in which an electron is lost, either completely or even only partially, is considered an oxidation reaction. So oxygen is not even involved here, but that's considered an oxidation reaction. Later on, we'll talk about reactions where oxygen is involved, and things will become maybe clearer, maybe a little bit more muddled. But the definition that you should know is that losing an electron, the process of losing an electron, that is oxidation. On the other hand, gaining an electron, so chlorine is gaining an electron here, that process is called reduction. This is a reduction reaction. This is an oxidation reaction. They never occur separate from each other. You can't just have one and not the other. So when I took chemistry, my chemistry professor at this point would take out his tape recorder, I think it was, and he would play us this Frank Sinatra song that I'm not going to play for you. And it's this song that's called Love and Marriage, which then goes on, says, goes together like a horse and carriage. This, believe me, brother, one never goes out without the other, or something like that. So the point here in this playing this song for us was to bring home the idea that something always goes together. And in the song, it's love and marriage. Of course, we're not going to talk about that and whether that really always goes together. But in chemistry, that does go together. So if you remember anything from this little offshoot here, this little reminiscence of my own days when I took chemistry, is that in chemistry, for sure, oxygen and oxidation and reduction always go together, right? You can listen to that song if you want. Maybe that helps you remember this. But never forget, oxidation and reduction reactions go together. So they're coupled. And there's another way, how you can remember that, and that is if you just remember the term oil rig, maybe that works for you. That tells you that oxidation is losing, OIL, right? Oxidation is losing. Rig, reduction is gaining. So oil rig, oxidation is losing, reduction is gaining. That's how you can remember what an oxidation or what a reduction reaction is. So it looks pretty straightforward in this example. Let's look at one that's not quite so straightforward. So if you look at methane and you look at oxygen, and you see that this is burned, so this is not a complete chemical reaction, as you see. There's no oxygen on this side, there's no hydrogen on this side. But if you produce CO2 from methane, then you also have a redox reaction, oxidation and reduction reaction. And the way that you can look at it is you can see here that the CH4, that carbon becomes CO2. So that loses electrons. Hard to see, right? There's nothing that becomes positive or negative. So hard to see here. And the same with oxygen. Why is this gaining electrons? So the way that you can see that is by looking at electronegativity differences. So here, if you look at the electronegativity differences between C and H versus C and O, I wrote them down over here, but you could calculate them on your own. Difference here is 0.3, and here it's 1.0. So that means that electrons close to the carbon have moved further towards the oxygen. So here, see, this is not very polar, right? Hardly polar at all. So the electrons are almost equally distributed between the carbon and the hydrogen. But here, that is a different story. Which one is the more electronegative element here? Carbon or oxygen? Well, it's oxygen, right? Oxygen is further on the right in the periodic chart. Not very far, but it is a little bit further over to the right. So the electrons are moving towards the oxygen. And that means, if you look at, you calculate the difference, this now has become a more polar bond. That means the electrons have moved away from the carbon. And that means the carbon has lost some electrons. Not completely, but it has lost sort of the electrons from its sphere. While the oxygen that has now become water, that this is a carbon dioxide. So, but if we look at the water here, the oxygen molecule in the water, there you have the movement of electrons towards it, right here. In this case, I have this actually written down for you. If you look at oxygen and water, oxygen, of course, there's no difference. Oxygen, hydrogen, there's a difference of 1.4. That means oxygen now has gained strong influence over some electrons from the hydrogen that are moving towards the oxygen. And so therefore, that oxygen here has gained electrons and therefore has become reduced. So there's a tip that I give you on how to look at this from the biologist's point of view, if you will. So if you look at oxidation and reduction reactions, they're almost always organic in nature when a biologist looks at it. And for me, a better way of remembering or quickly seeing what's going on here is, rather than looking at the electronegativity and calculating this out, is to just look to see what hydrogen does. Because in almost all cases, in organic biology, organic chemistry, biochemistry, if you will, we're looking to see whether or not hydrogen has changed its place. And here's my tip. Just look at this. Often the gain of an electron is better seen by the addition of a hydrogen, because the hydrogen brings electrons with it and doesn't hold onto it very strongly. So this part here is not even that important. But important is that if you add hydrogens, that almost always is a reduction. So look at this here. Here you have added hydrogens. So the act of gaining hydrogens, adding hydrogens, that is a reduction reaction. So then the other one has to be an oxidation reaction. In many, many cases, that also means there's an addition of oxygen. But the addition of hydrogen is actually an easier way to see which one is the reduction reaction. All right, so we've looked at redox reactions. We've looked at, we've defined it as the loss and gain of electrons. We've looked at electronegativity as the ultimate reason for why electrons move partners, and how we can determine whether or not something has been reduced or oxidized. And then I gave you this tip that you could just look at the hydrogens, and in almost all cases, you'll be right with that. So let's go back to your book and let's see how your book describes this, because you're going to read this in your book, and then you might as well see how they describe it. So they say there's an oxidation and a reduction reaction, and they want to show you this with the example of glucose. And they say that between the carbons here, carbon atoms share electrons equally. And now, after the reaction to carbon dioxide, they shade this here a little lighter brown, a little darker brown, and they say the dark shading represents the increased electron densities. In other words, electrons have moved over to the oxygen, of course, because of the electronegativity. That's what it says down here as well. So that then is an oxidation reaction. And in the reduction reaction examples, they show oxygen sharing the electrons equally. And then here you see the darker brown shade on this side. So now electrons are more closely moving towards the oxygen in this OH bond, where water is the example. So why not just think about it here as reduction is the addition of a hydrogen. Here you see that. Oxidation is the addition of an oxygen. Here you see that. But it's easier and fits better in most cases if you just look at the hydrogen. So yeah, don't tell a chemist that that's how I define redox reactions. But as a matter of principle, almost always you will be correct if you look at the movement of hydrogens. Okay, so we still really don't understand where the energy is contained as the electrons are moving around, right? We now see how electrons are moving from one place to another, and how that shifts the energy that is contained in these electrons. But there's, in biology, a particular compound. There's a few compounds, but there's one that we'll start looking at, and that might be the most important one for right now, that's considered an electron carrier. There are multiple types of electron carriers, but there's one particular electron carrier that is very frequently used in metabolism, and that is called NADH, or NAD, nicotine amide, and it's called nicotine amide dinucleotide. Just let for right now, let us call it NAD. So I say here in organic reactions, electrons can be transferred via an electron carrier molecule. More important than trying to understand what this long, big molecule, this formula here all holds, is that it can carry electrons by taking hydrogens together with their electrons. So I say here in metabolism, hydrogen is extracted from food molecules. So this is hydrogen that comes from food molecules. We'll see exactly how. And when I say these electrons can be carried, where can they be carried? Well, this is the molecule here. Now let's look at this a little bit and see what it is. This part down here, if I just sort of circle this part here, you should already know, you should already recognize what it looks like. What does it look like here? You have a phosphate, you have a sugar, and you have this base. So this looks sort of like DNA, right? Like a molecule that could be part of DNA. That's why the whole name here, nicotinamide dinucleotide, has nucleotide in its name. Because this is a nucleotide. This is almost a nucleotide, except that this base up here is not one that we use in DNA. It's a nicotinamide molecule. Not so important what this is called. What's important is that it is positively charged here at this nitrogen. Remember, nitrogen has 5 electrons, and here has 1. There's 2 electrons here, 1 belongs down here, 1 belongs up here. So I could say 1 electron here, 2, 3, 4, right, that are around the nitrogen. Nitrogen has 5 normally, now it only has 4, minus 1 negative charge leaves you with 1 positive charge. So that's our positive charged nitrogen here. So NAD plus 2 hydrogens, and these are not protons, these are not charged, these bring their electrons along. Where that goes, that hydrogen, is right here. So that can attach the hydrogens with the electrons, and it kind of looks like this. So before it looks like this, afterwards it looks like this. See, there's 2 electrons in this bond. Now there is an extra H and 2 electrons, traditional electrons in this bond. So you go from NAD plus, here is a positive charge, to just no charge because you bring an extra electron to balance that charge. And now I'm going to ask you which one is the oxidized form and which one is the reduced form. So look at this for a second, maybe pause me, think it through. Is this the oxidized form or is this the oxidized form? Which one's the reduced, which one's the oxidized form? All right, so you thought about this for a while. Well, this is the oxidized form and this is the reduced form. And maybe you have looked at your periodic chart, maybe you've thought about carbon here, maybe you've thought about hydrogen and electronegativity. But if you just think about addition of hydrogen, that's all really you need to know that this is the reduced form. It has an H right here in its sum formula, NADH. That's the reduced form, that's the oxidized form. All right, so that is an electron carrier. That is an electron carrier because it carries the electrons here, and this reaction can be made, can be reversed. So you can bring in the electron basically by using this hydrogen that brings in 2 electrons, and so now there are 2 hydrogens here, and that's why it's called an electron carrier. Why is it this particular structure? That's really not a question I can answer. I'm not sure if it's answerable. It's just a molecule that has the ability to do this. Electron transfer is needed in the cell in order to burn sugar in small incremental steps to not release all the energy in one step, which would burn the cell. So we want incremental steps. Incremental steps can be had by using this electron carrier. This was around already because it's almost like DNA. So in evolutionary history, the next step to the formation, the evolution of this kind of molecule that has the ability to carry the electrons might have been relatively easy to achieve. And so anything that works well, of course, in evolution is retained in evolution. And so that's the reason why this molecule is the molecule that is carrying so much of the electrons, because it works. And so therefore, evolution has retained it rather than changed it. Okay, so now we've talked about the chemistry, we've talked about some sort of principles about chemistry, we've talked about redox reactions, and we're going to see how these redox reactions are all involved in these cellular respiration steps. Now, we're moving in from the 30,000 foot level view at the very beginning, where we just said that sugar gets broken down and energy is released, to the next level where we are looking at the individual steps, and we're going to give them some names, and then we're going to look at each of these individual names and look at the steps that make up these larger steps. So your book calls them stage 1 through 4. Oftentimes they are described or discussed as 3 major steps because these 2 are sort of put together. Let's call them 3 or 4. The point is that you know what these 4 steps are. Okay, so there's glycolysis first. That is the breakdown from glucose to a molecule that's called pyruvate. I'll show you the structure in a little bit. I don't expect that you can write down the chemical structure for pyruvate, not for this class. Having said that, if you're a bio major, you're going to take biochemistry at some point in some form. Most likely, you'll have organic chemistry. You also have cell biology. So you will at some point have to learn some of these structures. I will tell you what for this class is important for you to remember. And that is not that you write down the structure for pyruvate. So in these stages, fuel molecules, sugar, are partially broken down, producing ATP and electron carriers. So I write over here as something that you should remember. Output of the first step that's called glycolysis and pyruvate oxidation, so I'm just moving, putting them together here, is ATP, a small amount of ATP, I say, and that's because relative speaking to this step, which releases a lot of ATP, this is a meager amount of ATP, but it's some. And we call this ATP via substrate level phosphorylation. Substrate level just means that the substrate is, remember from enzymes, that enzymes work on something, and what they work on, that's called the substrate. Well, the substrate here is the sugar, and the ATP gets released directly from the sugar. And so that's why it's called a substrate level phosphorylation. Phosphorylation, of course, means the movement of phosphate from a stage where it's not bound to ADP to ATP, diphosphate to triphosphate. So you're adding a phosphate. That's phosphorylation. That's what that step, what that term means. And then we're also producing these electron carriers. I showed you an electron carrier, NADH, NAD to NADH, just before this. And then carbon dioxide is also released. So you should know what's happening during glycolysis and pyruvate oxidation in terms of ATP, electron carriers, and carbon dioxide. And then if we move to the citric acid cycle, you see here that this is a linear pathway. We'll look at the individual steps. You'll see better why that's a linear pathway. That molecule is then fed into something that's called the citric acid cycle. And it's a cycle because molecules get sort of recycled where stuff comes off, stuff is added. So in this case, a molecule, it's called acetyl-CoA, is added. But carbons are broken off in the form of carbon dioxide. But then in the end, carbons get added again. So this is a constant change between just a handful, a few more molecules that are cycling around. That's why it's called a cycle. There are multiple cycles in biological metabolic reactions. This is the first that you might learn about. We'll go into detail. This is the citric acid cycle stage, and in this stage, just as overview, these fuel molecules that are originally broken down in glucose, they are now fully broken down, producing a little bit more ATP and a few more electron carriers. And again, this happens via substrate level phosphorylation. So small amount of ATP, some CO2, and electron carriers, quite a few electron carriers. These electron carriers are then, they're siphoned off from this reaction. You see how they're siphoned to here. And these electron carriers are then fed into the last step of cellular respiration, stage 4, which we call oxidative phosphorylation. So here we have substrate level phosphorylation. We don't need oxygen. We just need the substrate, sugar. Here we don't need a substrate really. We just need the electrons that come from the electron transport chain. And they're transported via these electron transporters, right? NAD, for example. NADH, I should say, in the reduced form. And here we need oxygen. So that's why it's called oxidative phosphorylation, because now you're adding phosphate, but you absolutely need oxygen for that. So this will not occur unless oxygen is around. And the waste product is water. And of course, that is the water that we exhale. So when we breathe out the carbon dioxide and the water, this is where the water comes from the electron transport chain. And in the electron transport chain, a ton of ATP is made, not through substrate level phosphorylation, through a very different mechanism involving a membrane carrier. So we will come back to how things can move through membranes very shortly when we talk about how ATP is made in oxidative phosphorylation. That happens in the next lecture. So today we're going to talk about these first 3 stages: glycolysis, pyruvate oxidation, and the citric acid cycle. All right, so if we look at this figure from your book, and we compare it with a figure that we already discussed earlier in the lecture, I just want to sort of connect these two for you in your reading. So we have, I say here, stepwise input and release of energy during glycolysis and citric acid cycle is shown in here, as it is shown here. So previously I told you when you burn sugar, which we watched in the YouTube video, you get all that energy released in one step, essentially. Here we have all these individual steps, and of course these individual steps are chemical reactions that occur during glycolysis, pyruvate oxidation, and the citric acid cycle. And in each of these steps, you'll see that here's our energy level. So look at this, just like this. This is our reaction scheme. We have a exergonic reaction, right? Multiple exergonic reactions, each of them requiring a little bit of activation energy. And you can see here that this each of these bumps here, the activation energy is this increase in energy that you see here on the y-axis. So there's an increase in free energy, and you see that by going from negative to less negative. So that's why it's an increase. And we have the increase because we give ATP in there, right? So that is our activation energy. And then at some point we release energy. We're getting off ATP and NADH. We release more of this. We're getting more ATP. We release more, and we're getting NADH this time. This is the stage where we're moving from glycolysis to citric acid cycle. That's the molecule that's called pyruvate. That's in between those two. That's why it's a special one. That's why it sometimes gets its own step. And then during the citric acid cycle, you see the formation of NADH, more NADH. There's a little bit of ATP coming off here, more NADH. And then there's this other one that's called FADH2. That's also an electron carrier. That's the one where I sort of said earlier it's a little bit less important. It's really just, it's also important, but if you understand NADH, then you also understand FADH. Similar in principle, carries electrons. Okay, so what you should take away from this is that this is a stepwise input of energy and output of energy during these two pathways. And we're producing ATP, we're adding ATP at the beginning, but we're getting a bunch of NADH. And later on you will see how NADHs, each of these NADHs can produce a ton of ATP. All right, so now let's go back here to the glycolysis and let's look at it a little bit more detail. Okay, so what your book does here is it shows you where we are, right? This is the scheme here on the left. Remember that we looked at glycolysis, we looked at the citric acid cycle, talked a little bit about the electron transport chain. I have not talked about fatty acids and amino acids here, and we will later on see how these can also be broken down and also release energy, and how they actually get funneled into the same pathway of the citric acid cycle. But that's not part of today's discussion. So this green arrow here is all of this together. So now we're really zooming in onto this part of the pathway. So, well, what's happening here? Let's look at this sort of step by step. There are three phases. We now call these phases. There's the preparatory phase where you use some ATP, and I earlier said that this means that you have activation energy. That's the use of the activation energy. Then you have the cleavage phase. So that is where the sugar gets cleaved into two different molecules. And then each of these two different molecules go through the same steps, but your book lists them as separate from each other, because now basically one carbon molecule of 6 carbons becomes 2 carbon molecules of 3 carbons each. 3 carbons in here, 3 carbons in here. You see that the numbers are the same, right? 6, 7, 8, 9, 10, it's the same here, versus up to here, the molecule was still 6 carbons, 1, 2, 3, 4, 5, 6. And then here's still 6, 1, 2, 3, 4, 5, 6. It just looks different now, right? It looks like it's a 5-member ring, but this is an oxygen, and here's an oxygen. So the carbons have just moved around a little bit with respect to their position to the oxygen. Here's a tip. What I would remember, what I would follow in these reactions when you have all these chemical names and that's all new to you, and maybe you're not really interested in learning all these chemical names. In fact, your book spares you the chemical formulas here, except for some that it considers to be important. What you should follow is the number of carbons. How many carbons are left? So here, 6 carbons are left. Up to here, 6 carbons are left. At this point, only 3 carbons are left, right? Here, only 3 carbons. Here, only 3 carbons are left. And then in the end, we are still left with 3 carbons. And then we want to see where those carbons go later on. So let me put here on the side what I think you should know from this reaction step, where glucose gets broken down, ATP gets added, NADH is added, NAD is added, and then NADH produced. Of course, here we have a reduction of NAD plus to NADH, which means that the rest, the molecule that's called glyceraldehyde 3-phosphate, has to be oxidized or reduced. Right? It has to be oxidized here because this gets reduced, so the other thing has to be oxidized to this molecule that's called 1,3-bisphosphoglycerate. I do not need you to learn all these names here. Here's what I need you to learn. This process called glycolysis starts with glucose, that's up here. It requires 2 ATP per glucose molecule, and we call that the activation energy. 1, 2. I don't care whether it's, you know that it's at step 1 and 3. All I know is what I care about is that you remember that it requires 2 ATP. Then what you also should know about this process is that one glucose molecule, which has 6 carbons, right? 1, 2, 3, 4, 5, 6. A 6-carbon sugar breaks into 2 molecules of 3 carbon sugars. So 2 3-carbon sugars, it's still 6 carbons. We haven't lost any carbons at this point. We've just broken some bonds. We have done some redox reactions. We have taken the energy. We have moved it to some electron carriers here. We've also created some ATP. So we've produced 4 ATPs and 2 NADHs, right? 1 here, 1 here. So 2 per molecule of 6. And in the end, we have a end product that you should know the name of, and it's called pyruvate, and it's still a 3-carbon compound. So that is the molecular structure here of pyruvate. Sometimes also referred to as pyruvic acid. So if you hear the term pyruvic acid, that's the same thing, except that it's in its protonated form. So the acid is called pyruvic acid. When the acid has given off its hydrogen and now no longer is a hydrogen-carrying acid, but is the anionic form. You see that here. This is where the hydrogen used to be, right? Where you now have an oxygen with a negative charge. Now it's called pyruvate. Pyruvic acid would have hydrogen right here and no negative charge. If you know this about glycolysis, that's great. That's good for Bio 111. All right, so glycolysis. Now the next step is the oxidation of pyruvate, and where that happens is important because all the other reactions downstream happen inside the mitochondrion. This happens in the cytosol, so it's not in the mitochondrion. It happens outside. At some point, this molecule needs to be moved into the mitochondrion where all that energy, remember that we said mitochondria are the place where most of the energy of the cell is produced. So this molecule pyruvate that comes from the glucose has to be moved into the mitochondrion. That happens in this next step. So let's remember what the mitochondrion is. I say here glycolysis happens in the cytosol. Sometimes that's also referred to as cytoplasm, cytosol. You hear me use both of those terms. And the citric acid cycle happens inside the mitochondria. So inside the mitochondria have an outer membrane and an inner membrane. And in between the outer and the inner membrane, there's the intermembrane space. And then inside the second membrane is what's called the matrix. So these are terms that you later on need to know because this membrane here and the movements of hydrogens mostly hydrogens and electrons, around this membrane is what's producing a lot of the energy. So it's really membrane transport that creates energy, and that will bring us back to the lecture that we had when we discussed membrane transport. So here's a membrane that we want to pay some attention to later on. But for right now, we just have to bring in the molecule from the outside. We're not doing anything along the membrane yet. We're just bringing in that molecule that's called pyruvate. So maybe you want to take a break here. You have talked for 50 minutes. Take a little break here. Come back in a little while, and then we'll bring in the pyruvate, and we do the tricyclic acid cycle, or the citric acid cycle. All right, so here we are. Now we want to take in our pyruvate. So just as an orientation where we are, glycolysis happened, glucose happened, pyruvate was the last step. Some ATP was produced. The pyruvate is now brought into the mitochondria. So here's a mitochondrion in the blow-up, right? This is the mitochondrion. All of this here. This is the membrane, the outer membrane and the inner membrane, and this is the intermembrane space. Pyruvate, 3-carbon compound, right? Has to be moved into the mitochondrion. As this is happening, there's one reaction, and a lot of things happen in this one reaction. That's why it gets its own step. So pyruvate is oxidized. You already know that because the whole step is called pyruvate oxidation. And in the end, we get another molecule whose name I would recommend that you know. And it's called acetyl-CoA. Why is it called acetyl-CoA? CoA because it has it's bound now to something that's called coenzyme A. Just out of laziness, it's called CoA rather than coenzyme A here. That's a very large molecule. We don't need to look at the structure. What coenzyme A does is it activates molecules. It sort of gives it a package of energy, like a battery, if you will, of energy that it sticks onto molecules, not just acetyl, but also other molecules in the cell in other types of metabolism. But in this case, let's just think about it as this battery pack that gets stuck onto this part, the leftover of pyruvate. So this part here becomes this part here. Then these parts come off, and you see there's a C and two O's, CO2, right? So that comes off, and that's what goes here. So now we have CO2 coming off of this molecule. This molecule is broken. That part gets stuck to this coenzyme A battery package. Now we have supercharged acetyl. And why is it called acetyl? Because acetic acid, vinegar essentially, is what this would be if the CoA wasn't there and you had a hydroxyl group. So if you had the carboxyl group here, COOH, with one methyl group at the end, one CH3, if that was COOH, then you would have acetic acid. If this was COOH, right? So that's why it's called acetyl-CoA. The third thing that happens at the same time is that NAD plus becomes NADH, and then there's usually this H plus around. So NADH, that's what we need to discuss here. So NADH is what we have. If you think about this, in this entire reaction here, combined reaction, right there, they're coupled with each other. Where's our redox parts, the reduction and the oxidation? Well, what gets reduced here? What gets oxidized? Is this a reduction or an oxidation reaction? From here to here, is this being reduced or is it being oxidized? Well, it's being reduced because you remember, easiest way to look at it is to just see where the hydrogens go. There's an addition of a hydrogen, so therefore it must be a reduction reaction. Therefore, this part here must be oxidized. That's a little harder to see, you know, addition of oxygen, I told you, not so great here as a way to see that, right? You could look at movement of electrons, you could get out your periodic chart, you could look at electronegativity, and you would figure it out. But it's just so much easier to just say, okay, there's an addition of a hydrogen here. This must be the reduction reaction. This must be the oxidation reaction. Pyruvate gets oxidized. It becomes coacetyl A. NADH is produced. Carbon dioxide is released. That's what we exhale, right? Water and carbon dioxide. And then coenzyme A is required for the battery package. You will see that this comes off pretty quickly after the next step. So that is what happens during the pyruvate oxidation. So let me put down here what I think you should remember. You should remember that this step starts with pyruvate. Pyruvate enters the mitochondrion. One carbon dioxide is split off. One NADH is produced. The endpoint is acetyl-CoA. This is its C2 compound, 1, 2 carbons. This is its C3 compound, 1, 2, 3. Where's the carbon going from 3 to 2? Well, of course, it goes here, right? 1 C is left over here. So that's where carbon gets lost from the system. Okay, third and last step for today is the citric acid cycle. So here's our sort of overview. We started with glucose. We broke it down in glycolysis. We produced a little bit of ATP through substrate level phosphorylation. We made pyruvate. Pyruvate now enters the mitochondrion. As it's doing so, it releases some NADH. It produces carbon dioxide, and it becomes acetyl-CoA. Acetyl-CoA is now the beginning, the entering point for the citric acid cycle. Here's our citric acid cycle. Here's acetyl-CoA. That goes into the citric acid cycle. So this is how this pathway is connected to this pathway through acetyl-CoA. And I told you this battery pack here is added temporarily. It charges, it supercharges, whatever you want to call it, this bit of this molecule. And then you see in the next reaction, in the next step, it already comes off. Already comes off here as this CoA SH. That's the form in which it normally floats around in the cell and then is reactive to produce the next supercharged molecule, to charge it up to become, I don't mean charged in terms of electron, like with negative charges or positive charges or anything like that. Just sort of, it becomes active. That requires some water from the surrounding. That's all not so important. What is important is that this bit here, the 2 carbons with that oxygen, gets put on top of an acceptor molecule that is called oxaloacetate. So here is the structure of oxaloacetate. You don't really need to be able to write that down for this class. But see, what you should know is that it has 4 carbons. And it actually says this here, 4C. 4 carbons, 1, 2, 3, 4. Maybe you already see also that it has these carboxyl groups that are protonated, so that are deprotonated. So no proton on it, therefore hydrogen is off. Left its electron, so it's negatively charged. So whenever you see COO, that is the remnants of the carboxyl group. So here's a carboxyl group, here's a carboxyl group that is deprotonated. And so carboxyl groups come from organic acids, right? So this was an acid at some point. And that is why this is called the citric acid cycle, because the first step that is made from the fusion of oxaloacetate and acetyl-CoA is this part here gets stuck on top of this. See how that is moved on top here, so that you now have 1, 2, 3 carbons. And then you have 3 more carbons, and each of those are carboxyl groups. So this is an acid essentially, and that's why this compound is called citric acid. That's actually the stuff that makes lemons so tart, so acidic, so sour. And why sometimes you also see this this cycle called the tricyclic acid cycle. So TCA cycle or it's also sometimes referred to as the Krebs cycle. Krebs cycle. Krebs was the guy, Hans Krebs. We call it Krebs here, him. But Hans Krebs was the guy who worked on this, figured this all out. This is almost 100 years ago. Hard to imagine how long ago this is when this was all figured out, each of these steps. Anyway, so the first step, this gets stuck on this. We are releasing the coenzyme A. We now have citrate. And at that point, the citrate moves through a number of different steps. And each of these steps, something happens. Either the molecule just gets sort of shifted around, looks a little different afterwards, or it actually gets changed in such a way that carbon comes off. So that means it becomes smaller, right? At the beginning, it has 6 carbons. In the end, it has 4 carbons. Where do those 2 carbons go? Well, they go here. First carbon comes off here in the form of carbon dioxide. That's the carbon dioxide that we exhale. Second carbon comes off here, carbon dioxide that we exhale. So from a molecule with 6 carbons, 1, 2, 3, 4, 5, 6, we remove 2 carbons and we end up with a molecule which only has 4 carbons. That is called oxaloacetate. And that is also referred to as the receptor molecule of the citric acid cycle. Well, what else happens in this pathway? NADH is produced here, NADH is produced here, and NADH is produced over here. Those are 3 NADH molecules that are produced for each round of the citric acid cycle. So when I say a round of citric acid cycle, that doesn't mean that the one sugar molecule with 6 carbons gets completely broken down, right? Because you see that the only thing that was added was actually 2 carbons, and only 2 carbons get used here. So there's only 2 carbons that are required for making the cycle go around once and producing each of these molecules. Then there's another 2 carbons that can be used, and they can be broken down after 2 more carbons from glycolysis were added. So with every step of this, with every turn of the cycle, we're producing 3 NADHs, we're producing ATP over here through an intermediary step, through GTP, not so important. And then we're producing also this FADH. So that's also an electron carrier. So, okay, what should you remember of all of that? What I think you should remember is that you should remember that this is sometimes referred to as the Krebs cycle, the TCA cycle, or the citric acid cycle. So that starts with acetyl-CoA, which comes from glycolysis, and then it's a 2C compound. So if you can, just as I said earlier, if you just sort of count carbons, that's already really good, because then you see where carbons go, and how the 6 carbons in the sugar end up all being in single carbon dioxides. So starts with a 2C compound and an oxaloacetate, which is a 4C compound, producing a 6 carbon compound. The first product is citrate. You should know that. You should know that 2 carbon dioxides are split off here and here. Where exactly, I don't care so much that you know. And you should know that the cycle itself produces 3 NADHs, 1 FADH, 2, and 1 ATP. And that the end product is the beginning product, if you will. It's oxaloacetate. This product is regenerated after it's modified to citrate by going through this entire cycle here. So if you remember this, I think you're pretty good off. One more thing before I let you go for today about the citric acid cycle, and that is that while you might think, oh, this is just one metabolism, one step of metabolism, and a lot to learn if you want to learn everything about metabolism in humans, animals, and plants as well. Well, what's nice to know might be that the citric acid cycle is so common in so many metabolic pathways that once you learn it, you learn many of the other pathways, at least parts of it as well, because all of these other compounds that are made in organisms, we use the same steps of the citric acid cycle and the pyruvate oxidation. And so what they are is pyruvate can go the other direction. It can actually produce sugars in a pathway, a series of steps that produces sugars. It can produce amino acids. So we haven't really learned all the names of the amino acids, but you may have seen them before. So alanine, you might already recognize as one of the amino acids. Glutamine, glutamate, other amino acids over here, aspartic acid or aspartate. Those are all different types of amino acids that can be made in organisms when they don't have them. So if they don't pick up these amino acids through food, for example, then the body can produce them through the citric acid cycle, through breaking down sugar and then turning that sugar into amino acids and then essentially into proteins. Here from the acetyl-CoA, that can go into the citric acid cycle, but it can also produce lipids. So you may have heard, if you eat a lot of sugars, you get fat. Well, where does the fat come from if you eat just sugar? Well, it comes from exactly this pathway. Sugars get broken down and then they get metabolized. If the energy isn't needed, then the extra sugar can be stored as lipids. So the fat can be used as a storage form of energy. Here we can, out of intermediates of these steps in the citric acid cycle, we can make heme and chlorophyll. Heme, of course, is part of the hemoglobin of the blood, and chlorophyll is what captures energy from the sun. We'll talk about chlorophyll and photosynthesis in a chapter later on in a series of lectures. And then here we have aspartates, pyrimidines, and over here are purines. Those are substances, purines and pyrimidines, the different types of bases that make up DNA, nucleic acids. So you can make lipids, you can make amino acids, you can make nucleotides, all with this particular set of pathways from sugars. And that's why this pathway is so important to know parts of. What you should know is what I told you along the way. Let's summarize this. From this lecture, maybe you want to remember that cellular respiration breaks down sugar and produces ATP and carbon dioxide, and it requires oxygen. We talked about redox reactions. We said that they're coupled. They move electrons between reaction partners. We said that in cells, electrons can be moved to electron carriers, which can then help transfer these electrons and hydrogen. Cellular respiration involves 4 major steps, and I just went through 3 of those: glycolysis, pyruvate oxidation, and the citric acid cycle. And then for next time, we will talk about oxidative phosphorylation. And then each step of these produces or uses, you should look at each of the steps: ATP, NADH, carbon dioxide, and oxygen. Oxygen only comes in later, and that comes into play when we talk about oxidative phosphorylation. You should memorize this for each of the 4 steps. So where do these 4 types of compounds go and come from? How are they used and produced? And citric acid cycle is used in many metabolic pathways, not just in cellular respiration. Okay, with that, I leave you, and I'll see you in class. Bye-bye.